Have you ever wondered how traits are passed down from parents to offspring? Why you look similar yet different from your parents? This is the realm of genetics, the science of heredity and variation. At the heart of genetics is deoxyribonucleic acid, or DNA, an intricate molecule discovered in the mid-20th century. From dictating your eye color to predisposing you to certain health conditions, DNA plays a key role in heredity. The answers to these questions lie in the fascinating field of genetics. Our journey begins in the mid-1800s, a time when the structure of DNA was still a mystery. The first significant step towards unveiling this enigma was taken by the Swiss biologist Friedrich Miescher. In the late 1860s, Miescher isolated a substance from the nuclei of white blood cells, which he called nuclein, which we now know as DNA. Fast forward to the 1950s, and the baton of discovery passed to Rosalind Franklin, a British scientist. Franklin used a technique called X-ray diffraction to take images of DNA. One of her photographs, famously known as Photograph 51, provided key insights into the structure of DNA. However, Franklin's contribution often goes unnoticed as she passed away before the Nobel Prize was awarded. Enter James Watson and Francis Crick. These two scientists, working at the University of Cambridge, used Franklin's Photograph 51, along with other data, to propose the double helix model of DNA in 1953. The model suggested that DNA is made up of two strands that twist around each other like a ladder, with the rungs composed of pairs of 4 types of nitrogen bases. Their landmark paper in the scientific journal, Nature, revolutionized our understanding of genetics. For their work, Watson, Crick, and another scientist, Maurice Wilkins, who also contributed to the DNA structure research, were awarded the Nobel Prize in Physiology or Medicine in 1962. This discovery was a game-changer. It not only explained how DNA can carry genetic information, but also how it can replicate itself, a mechanism essential for life to exist and evolve. It was the missing piece that helped us understand the complex puzzle of life on a molecular level. And thus, the double helix structure of DNA was discovered, opening up a whole new world of possibilities. This discovery laid the foundation for modern genetics, and has since then been instrumental in various breakthroughs in the field of biology, from genetic engineering to personalized medicine. The DNA molecule, a beautiful double helix, is more than just a pretty structure. It is a masterful piece of biological architecture designed with a purpose. If we delve deeper into this structure, we'll find that DNA is composed of 2 long chains of nucleotides. Each nucleotide consists of a sugar, a phosphate, and one of 4 bases: adenine, cytosine, guanine, or thymine. Adenine pairs with thymine, and cytosine pairs with guanine, creating a perfect harmony of base pairs, like love letters in a timeless genetic language. Now imagine this marvelous structure needs to replicate to make a copy of itself. How does it do it? DNA replication is a sophisticated process. It begins with the unwinding of the double helix by an enzyme known as DNA helicase. This creates a replication fork, where DNA polymerase can attach and start adding complementary nucleotides to each strand, following the rule of base pairing. This process continues until 2 identical DNA molecules are formed from the original one. It's like a flawless choreography, where every step is meticulously coordinated. But DNA doesn't stop there. It has more stories to tell, more tasks to fulfill. It also needs to transcribe its genetic information into RNA in a process called transcription. Here, another enzyme, RNA polymerase, comes into play. It opens the DNA at the required gene and synthesizes a strand of RNA, with uracil replacing thymine. This RNA will later be translated into proteins, the workhorses of our cells. So, the DNA molecule, in its elegant double helix, is not merely a static structure. It is dynamic, constantly replicating and transcribing, ensuring our genetic information is accurately passed on. It is the central hub of life, a testament to the beauty and complexity of biology. So, our genetic information is safely stored and accurately transcribed, thanks to the unique structure of DNA. Genes carry the instructions for making proteins, but how are these instructions read and followed? Well, It all begins with the genetic code, a system that enables these 4 nucleotides to code for the amino acid sequences in proteins. It's like the language of life, where words are made of 3 DNA bases called codons. Each codon corresponds to one of the 20 amino acids used in proteins, or it signals to start or stop protein synthesis. Now, to translate this code into proteins, we need the help of messenger RNA, or mRNA. This molecule is a copy of a segment of DNA made during a process known as transcription. This mRNA then carries the genetic information from the DNA in the nucleus to the cytoplasm of the cell where proteins are made. Enter transfer RNA or tRNA, the interpreter of the genetic code. Each tRNA molecule has a specific amino acid attached to it and a unique set of 3 bases, known as an anticodon, at one end. This anticodon can pair with a codon on the mRNA. But where does this pairing happen? Inside the cell's protein factories, the ribosomes. Ribosomes read the mRNA, and for each codon, they bring in the tRNA with the matching anticodon. As the ribosome moves along the mRNA, it links the amino acids carried by the tRNAs together, creating a chain. This chain folds into a complex 3D shape to form a protein. So let's put it all together. Your DNA holds the blueprint for all your proteins. This information is transcribed into mRNA, which carries it to the ribosome. The ribosome then uses tRNA to translate the mRNA's message into an amino acid chain, which folds into a protein. This process of translation is the final step in the central dogma of molecular biology: DNA to RNA to protein. It's the way your genes are expressed, the way the information in your DNA is used to do the work in your cells. And voila! The genetic code hidden in our DNA is translated into functional proteins. How are traits passed from one generation to the next? The answer lies in the laws of heredity. Delving deeper into the molecular basis of heredity, we encounter the principles of Mendelian genetics. This fascinating concept was proposed by Gregor Mendel, a monk with a keen interest in pea plants. His curiosity led him to discover the fundamental laws of inheritance, which form the bedrock of genetics. Mendel's laws are twofold. First, we have the law of segregation. This law states that during the formation of reproductive cells, pairs of hereditary factors, or genes, get separated. This means that each offspring acquires one factor from each parent, shaping the inherited traits. The second law, the law of independent assortment, adds another layer of complexity. This law asserts that genes for different traits assort independently of one another in the formation of gametes. In simpler terms, the gene you inherit for hair color doesn't determine the gene you'll inherit for eye color. They are independently assorted. These principles of Mendelian genetics play a crucial role in heredity. They explain how traits are passed down through generations and why each of us is a unique blend of our parents' traits. They provide the blueprint for diversity, dictating why siblings don't look exactly alike unless they're identical twins. Moreover, Mendel's laws are also the foundation for understanding genetic disorders. Some conditions, like cystic fibrosis or sickle cell anemia, are caused by specific combinations of genes inherited from parents. Understanding these principles allows scientists to predict the likelihood of these diseases appearing in future generations. It's incredible to think that the workings of our complex bodies and the continuation of life itself can be traced back to tiny molecular structures, and that these structures obediently follow laws discovered by a curious monk in his garden over a century ago. These simple yet powerful laws explain how traits are inherited, making you uniquely you. We've journeyed through the fascinating world of genetics, from the structure of DNA to the principles of heredity. We've traced the path of discovery, from the early breakthroughs that unveiled the double helix structure of DNA to the intricate processes of replication and transcription. We've explored how the genetic code is translated by RNA, acting as a blueprint for life. We've delved into the structure and function of proteins, the workhorses of our cells, and their role in carrying out the instructions encoded in our DNA. We've looked at the molecular basis of heredity and how the principles first laid out by Gregor Mendel in the 19th century still guide our understanding of genetics today. We've seen how these principles are at work in everything from human health to agriculture and conservation. The world of genetics is vast and complex, but it's also incredibly exciting. It holds the keys to understanding so many aspects of life, from how we inherit traits from our parents, to how diseases can be passed down through generations, to how we can use this knowledge to improve crops or save endangered species. So, here's to the beauty of genetics, to the curiosity that drives us to understand, and to the incredible discoveries that are still to come. Genetics truly is a fascinating field, holding the key to understanding life itself. Subscribe for more mind-blowing science content.